Blog Talk Radio. Welcome to another episode of M-Patient Myeloma Radio, a show that connects patients with myeloma researchers. There are two ways you can subscribe to M-Patient News: to hear about our previous episodes and learn more about our upcoming shows. Please go to www.mpatient.org on the homepage and sign up for our new M-Patient Minute, which is our weekly newsletter that contains all show information in one email. You can also subscribe to all posts as they come out. With the recent acquisition of Onyx by Amgen, I was curious to know how myeloma drug goes from an idea to a real therapy being used successfully in the clinic. We're very fortunate to hear from a top researcher, Dr. Craig Crews of the Crews Laboratory at Yale University, who achieved this very thing with his discovery and development of the drug carfilzomib for multiple myeloma. So thank you, Dr. Crews, for joining me today.
My pleasure.
Before we start, I'd like to give a short introduction of you for our listeners, if that's okay.
Please.
Dr. Crews is a Lewis B. Coleman Professor of Molecular, Cellular, and Developmental Biology in the Departments of Chemistry and Pharmacology at Yale University. He performed his undergraduate work at the University of Virginia in chemistry and earned his PhD in biochemistry at Harvard University. He is the recipient of the Senior Scholar Award at the Ellison Medical Foundation, continues as a visiting professor at a university in Germany, received the Bessel Research Award from the Alexander von Humboldt Foundation in Germany, and is a fellow of the Royal Society of Chemistry. Earlier in his career, he was also awarded the Donahue Foundation New Investigator Award and the Burroughs Wellcome Foundation New Investigator Award. He also serves on the editorial board of Chem Biochem and Chemistry and Biology. Dr. Crews has created multiple patents focusing on enzyme and proteasome inhibition. His drug carfilzomib, now called Kyprolis, was approved by the FDA for relapsed multiple myeloma in July of 2012. Dr. Crews, we all thank you for your deep research and for developing an improved therapy for multiple myeloma.
My pleasure.
So I guess the first question would be, how did you come to the idea for a new proteasome inhibitor for multiple myeloma?
Well, it was quite by accident. I was studying a compound from nature. It was a small molecule that was found in a microbial broth that had been discovered actually by a pharmaceutical company, Bristol Myers Squibb, in Tokyo. And they were interested in looking for novel anti-tumor agents. And they had found one that was quite promising, but they ultimately made a business decision not to explore it any further because they didn't know how it worked. And they anticipated that the FDA would want to know that information before they would approve any drug based on that natural product. And so they published it once they decided they weren't going to develop it any further. So I became aware of this molecule called epoximycin as a recently appointed assistant faculty member at Yale in the late 1990s when I was reading a journal article about this. And I became intrigued. It was a very potent compound without any idea of how it was working. And so I wanted to see if we might be able to help answer that question. What was the mechanism by which this compound from this bacterium could really kill tumor cells? And so I applied for a grant from the National Institutes of Health, NIH, and I was fortunate enough to get funding for this very basic question of how this compound affects tumor cells. And so I set out to do this, but the first stumbling block was we didn't have access to any of the compound because Bristol Myers Squibb had subsequently closed their research center in Tokyo, and I couldn't get my hands on any of the material. So my lab, which is half chemists and half biologists, we set out to actually make our own version of epoximycin. And so when we did that, which happened to be the very first reported synthesis of this compound, but this allowed us not only to study its effect and confirm its effect on tumor cells, but it also allowed us to start tweaking the structure of the natural product. And the first tweak we did was to basically put a handle, if you will, a molecular handle on the compound, and we went fishing. And by that I mean we would add it to cells, it would get inside cells, and we wanted, of course, to know how it worked, and that meant we had to find what protein was being bound by this, what protein was binding to this compound. And so when we fished out of the cell the protein bound to this compound, we found that it was the proteasome. And so for your listeners, the proteasome is a large cylinder, a very complex machinery inside the cell, whose job it is to degrade proteins. And so this was the first clue that our molecule could be working through blocking the function of the proteasome. It could be inhibiting the function of the proteasome because it was binding. And so at that point we published our results, and were quite excited about this because we knew from the work by Millennium of a small molecule that was working its way through the FDA approval process for multiple myeloma, and we know that compound today as Velcade or bortezomib.
Mm-hmm.
And so what we subsequently did, we asked ourselves, even though this was a potent molecule, now that we know what the target is of this molecule inside the cell, we asked ourselves if we could improve upon Mother Nature. And so I threw it back to the chemists in the lab and challenged them to tweak the compound structure a little bit here, a little bit there, and come up with something that was an improved version. And when they did that, after a fair bit of work, as you can imagine, we came up with a molecule that we call YU-101 for Yale University 101. And it was that molecule that we were able to demonstrate was many times better than the natural product from nature, epoximycin, and was many times better than Velcade, the compound that was soon to be approved for multiple myeloma. And so it was at that point that we confirmed that it was, yes, still a potent anti-tumor agent, and we were able to do that here at Yale. But that is as far as my lab felt comfortable in taking this project, because we really were able to take it from a very basic research question through to answering that question and then flipping it over to a more translational medicine effort to improve upon this, to make a lead compound, if you will, for further development as a potential therapeutic. But it was at that point that Yale University and myself and several other researchers started a company, and that company being called Proteolix. And so Proteolix took YU-101, added one last tweak to it to allow it to be more easily dissolvable in water, and took that into the clinic. And that molecule is today carfilzomib, or known as Kyprolis, as you mentioned.
You started this at what point? When were you getting the NIH grant, and how long did this process take?
So I got the NIH grant at the very beginning, and so the grant application is relatively straightforward in terms of the application. We pose a question, we explain how we're going to address that question, and then a team of our peers, scientists, serve on a review panel at the NIH to then pass judgment, if you will, on whether it should be funded. I should pause right here and just emphasize what I think is well known, but maybe it's not, and that is the funding for biomedical research in the U.S. has decreased in the last 10 to 15 years, to the point where for every 100 applications that are put into the NIH, 90 of them are being rejected. And it's clear that there are some that should be rejected, but as you can imagine, year after year after year, the bad ones, the mediocre scientists, they leave the field, and we're really cutting into bone now in terms of rejecting really good science. And that's not even addressing the question of making sure there's an funding environment that is supportive of young investigators like myself years ago when I was starting out and was given a chance. And so I encourage all of your listeners to be active supporters of not only the applied side, but also the basic research side of the U.S. biomedical community, because you never know where the next breakthrough drug is going to be coming from.
Right. And how, as patients, can we best do that?
Well, it's engaging your congressman or congresswoman, making sure that they know that this is a priority to you, making sure that they're aware that it's not always possible to predict what the best drug is going to be, and the importance for studying very, very basic things. Why on earth would, if you think about it, studying something that comes from some bacterium, some compound, and how it works is so far removed from ultimately the drug that was approved. But it's that type of very broad scientific inquiry that has set us apart as a country for decades from all the other countries, and has fueled the biotech industry, as well as the successes that we have seen in pharmaceutical development. And some might argue that the decreased number of new drugs in the pipeline today may be a reflection of the decreased investment that began, unfortunately, in the about 15 years ago.
Okay. Well, it is a challenge, and we've heard that from other myeloma researchers as well. And I think we'd like to know what we as patients can do. So those suggestions are great. So you've kind of taken us through the actual compound and then to when you pass it off at Proteolix. Can you give us an overview of what happened then, or what the other steps were to turn it into a drug? Sure.
So as many of your listeners may know, that the drug approval process has three phases. The first is phase 1 trials are safety trials, and are designed to determine what concentrations, how much of the drug can be safely administered to patients. And it's a very important clinical trial because we obviously want to be dosing at the safest concentrations possible, but it's oftentimes not always possible to be able to see an effect in phase 1 clinical trials. And so it requires a bit of altruism on patients' part to volunteer for phase 1. Once the amount of drug that can be safely administered to a patient is determined, then the next step being phase 2 clinical trials begin. And in phase 2 clinical trials, the FDA is asking the company to prove that their drug candidate is better than anything else that's out there. And the point of view from the agency is why should we allow another Me Too drug? It has to be better. And so these patients that are enrolled in phase 2 clinical trials will have already exhausted a series of other options, clinically speaking or therapeutically speaking, and therefore they're very challenging patients. And that's the point, is to set a high hurdle for any drug candidate coming out of phase 2. But if successful, if there is some hint that the drug is working in these patients that have been seen a lot of other therapeutics and have failed them, then that's when phase 3 and the final registrational trials, as they're called, begin. And they're called registrational because if successful, then one can register for an FDA approval based on those clinical results. And so in the phase 3, which are a much, much larger trial series that in terms of larger numbers of patients enrolled, this is where patients who have not had any prior or have limited prior exposure to other drugs. The idea is the FDA wants to see how this drug candidate behaves as if a patient had walked off the street into his or her doctor's office and been presented for the first time with a therapeutic option. So oftentimes these trials are run in a comparison with the standard of care, the best in class of another, whatever is currently the best out there. And so that way, but the point being that these patients would not be as
maybe not in an advanced of advanced disease stage, maybe.
Well, it's not they not so much advanced. It's not that they haven't seen other compounds and
or
so
other prior therapies.
Prior therapies, exactly.
Mm-hmm.
And so as I'm sure you know, prior therapies can change the course of the disease. The disease changes once it's been exposed to something else. It changes in response to these therapies. And so we wanted to, the FDA wants to have a head-to-head comparison with what's currently best in class and see how the candidate behaves. And so that's how things, so that's a phase 3. In the case of proteolics and carfilzomib, Kyprolis, because our phase 2 data were so convincing and so compelling, we actually got the company that acquired proteolics, that company being Onyx Pharmaceuticals, made the business decision to actually apply for FDA approval based on the phase 2 data, and they were successful. And so the FDA said, listen, given the limited number of options out there today, even though we haven't yet done all of the clinical trials, we will approve Kyprolis for those patients that are relapsed refractory patients. These are the patients who have failed other therapies. As I said, those are the patients that are recruited for phase 2. There are ongoing phase 3 clinical trials, and so if successful in those, then the company can apply for approval for what's known as frontline treatment, meaning that, as I said, naive patients that have just newly been diagnosed would be offered Kyprolis as their first option. But those data aren't yet available because the clinical trial is ongoing.
And right now you need to get a prior therapy, correct?
You have to have you have to have failed prior therapy, yes, for Kyprolis.
So, in total, how many trials really need to be run on a new drug like carfilzomib before it's FDA approved?
Well, in theory, three. Three, phase 1, phase 2, and phase 3. But in reality, there are, after a phase 1, which simply, as I said, sets the safety parameters, there are often multiple phase 2 and multiple phase 3. The idea being that you might want to try your candidate in combination with something else. And so each change in the design of the trial necessitates a new trial. And so if you want to couple Kyprolis with an IMiD class from Celgene, one of their molecules, to see if in combination it works better than the Celgene's compound by itself, then you need to do those types of studies. And so that's why it can be a bit confusing when a patient goes on clinicaltrials.gov, the website that lists all of the clinical trials ongoing today, searches for multiple myeloma or searches for Kyprolis. They would see a number of trials that are either wrapping up or are starting. And there are new indications, too. We've seen success, obviously, with multiple myeloma, but this particular drug, this proteasome inhibitor, Kyprolis, might have some efficacy, might be effective against, say, solid tumors. Mm-hmm. And so that necessitates another series of clinical trials.
Okay. And in terms of funding, so the NIH grant did the initial funding, and then when it was developed by Proteolix, how did they receive their funding? And then it kind of went to Onyx, who I'm assuming funds that research themselves. Or are there other funding steps along the way?
So the founding of a biotech traditionally involves recruiting of money, capital, from venture capitalists. And so these are investors who are willing to take the risk that they might strike it big or they might lose everything. And so the model has traditionally been, given the high failure rate in these companies, is that they're hoping that 1 out of every 8 or 1 out of every 10 companies that they start will hit it big, but the return, the amount of money they make on that one that is successful is more than 8 or 10 times the initial investment. So that's how they ultimately will make money, even though 9 out of 10 companies will fail. And so just to give you some numbers here, the company Proteolix was started with an initial investment of $18 million.
Okay.
And that allowed the tweaking of the structure of YU101 into the final candidate Kyprolis, as I mentioned, and also started funding safety trials, phase 1 trials. But subsequent trials required additional investments. And so those initial investors went out and talked to their friends and sometimes went back to their own firms and asking for additional money. The bottom line, if you will, is that by the time Onyx, 7 years later, bought Proteolix, the investment of all of the investors in Proteolix was over $140 million.
Wow.
Onyx, however, purchased that for what will be upwards of $850 million if the drug makes all of its clinical milestones. And so the investors made money, and they're now able to take those profits and go off and do this again with another 10 companies, hoping that one of them will be successful.
And similar. We would love for them to be similar.
Yes. Mm-hmm. And just to wrap up the story here on terms of the investment side, last week Onyx, having made that $850 million investment in proteolycs, was now sold itself for $10.4 billion. And it was purchased by the largest biotech in the world called Amgen. And so Amgen sees this successful multiple myeloma drug, and which is projected to be upwards of $2 billion a year in sales, and in a couple of years, and they wanted to have that in their portfolio.
Well, it's a huge success story, beginning with you. So we thank you, and one for patients as well. Now, I've heard researchers talk about what they call the valley of death for a drug. Can you tell us what that might be and how you overcame that with carfilzomib?
Right. So the valley of death refers to how far an academic can take, an academic researcher can take a project within a university setting, which is limited. We don't have the resources of a large pharmaceutical company. We don't have the animal testing oftentimes. We don't have the large team of medicinal chemists and whatnot and pharmacologists. And so there are a lot of discoveries that have great potential, but no one's picking them up as projects. And previously, the pharmaceutical companies internally had the bandwidth to be able to look at what is being done in academia, in the universities, and to say, that looks interesting, why don't we work on that for a little bit and see if we can make something of it. And so it was much more exploratory research. They were willing to take a project that was high risk and to de-risk it, if you will, kind of figure out, answer some of the obvious questions. And, for the most part, they didn't pan out, but some did. And that served as the very beginning of the pipeline, of the drug pipeline for these drug companies. What unfortunately has happened is with the mergers and acquisitions and all of the restructuring within the large pharmaceutical industry, the internal research and development has taken a hit, and a lot of the capacity to be able to evaluate very early discovery projects reported by academics like myself no longer is in place. And so they have turned their attention, the large pharmaceutical companies, instead of building it from within, they'll rather go out and purchase companies like proteolycs. And so what that means is that works for a while there still are companies like proteolycs, but if there's no one out starting the companies and there's no internal transfer from the university labs into large pharmaceutical labs at the very earliest stages, then the pipeline dries up. And so this valley of death, how do you take a project that's very promising but has a lot of obvious questions of whether it can be useful as a drug? How do you take that project from, say, my laboratory and get a large pharmaceutical company interested in it, where they are saying, well, that might be interesting, but we have all these concerns that need to be first addressed. And we're looking at ourselves on this side of the valley and say, well, we don't know, we don't have the capacity to do this. And so this is where biotech is stepping in, where the venture capitalists are saying, we're willing to assume the risk. There are questions, yes, but we see that if we answer those questions and successfully, then we might be able to turn around and sell this project or sell this company to a pharmaceutical company. And so, in a part, you might think, one way of saying is that large pharmaceutical companies are outsourcing, if you will, a lot of the innovation that they were originally had internally, but now they are outsourcing that. The challenge, of course, is how do we educate from my side, my fellow academics, the university professors, how do I educate them saying, listen, we need to take it to the next level. It's not sufficient. It's not enough just to discover something and publish it and hope that someone's going to pick it up. You have to champion this. You have to be an advocate for this. You have to, fight for what you think has great potential. And so, what I'm trying to do here at Yale and others and other universities are trying to make this transfer of technology from an academic lab to, say, a startup biotech easier, and that requires educating my fellow professors and saying, it's not that hard to attract people that are willing to invest in your story. And so I am very, very concerned about the Valley of Death, but I feel that I'm optimistic that we might be able to overcome this. The other thing that I do feel that I'm optimistic about is foundation-based venture philanthropy. So I know, for example, Polycystic Kidney Disease Foundation, PKD Foundation, the Cystic Fibrosis, and I think, and definitely the Leukemia Lymphoma Society. They are, these large foundations, as you may be aware, are taking a more aggressive stance to helping biotechs, not just the academic research, but allowing some of their funds to go to for-profit entities for the purposes of advancing drugs of their interest. My point is that this might be another entry, another way to address the Valley of Death. Is my point.
Yes, the LLS is very involved in helping support actual drug development. Like Gleevec is one of the examples there that was funded both by the NIH and the Leukemia and Lymphoma Society. The myeloma space also has its foundations, the IMF, the MMRF, and the Moore Foundation, who fund myeloma research and, in some cases, drug development. And now I have another question on a different topic. Now that you've run through the process successfully, what are the key lessons you've learned, and would you do anything differently the next go-around?
Yes. So key lessons. Well, the most important one is that it is possible. Oftentimes people don't start something because no one has gone before, and they think things aren't possible. And so I hope that one takeaway, at least for my colleagues, is that if there is something that is potentially a potential therapeutic that came from your laboratory, I would encourage people to find the necessary capital and team to start a biotech if they can't directly engage a large pharmaceutical company. So yes, I think that's the most important thing. I've been very fortunate in that this particular story was a fantastic one in the sense of the team at Proteolix were able to develop this drug in such record time, and now obviously it's approved and it is helping patients. The fortunate thing for me personally is that with this prior success, it's allowing me to more easily attract the investment in my next company, which I just recently have started this summer. We're still looking for what indications, but certainly multiple myeloma is one I know well. This is going to be an oncology-focused company.
Okay, well, this is a good next question then for you. My husband, when he talks sometimes, he quotes Bob Metcalf of 3Com, and he was the inventor of the Ethernet, and he said, most successful entrepreneurs I've met have no idea about the reasons for their success. My success was a mystery to me then and only a little less so now, which I think is a funny quote, but I think sometimes it's true in entrepreneurship anyway. So in science, is it similar? Or how can we replicate your success and the success of others when it comes to myeloma discoveries?
Well, that's a good point. I teach graduate students. These are people who are actually doing the work in a laboratory, training them to be future researchers, future professors. And one of the things that I try to teach, and I'm still grappling with this after 18 years, is how do you teach creativity? How do you teach innovation? And it's basically, I believe, it comes down to the ability to recognize opportunities. We are flooded, as you can imagine, every day with new pieces of information, and the vast majority of the information fit nicely into the preconceived models that we had constructed in our head as to how things should be working. Obviously, we have assumptions, models, and we test them, and that's the whole concept of experiments. But there are occasions when things don't work exactly as you anticipate that they are working, and it's those outliers. And you can imagine you could spend an extremely unproductive lifetime tracking down all of these outliers that might not be important. But I am trying to teach my laboratory how to recognize when an outlier is worth pursuing. And so that's something that I think is a lifelong lesson that if learned, could really have a major impact on the success of someone's career, whether that career is solely in academic research or if they have an interest in actually therapeutic development as a drug developer.
And I think that's an acquired skill, picking winners.
Yes.
And I think it's definitely a difficult skill. If it weren't, we would have a lot more successes.
Right, and so that's what I'm trying to do, is see if I can distill some of the skill set that I've developed over the years that to me feels natural, trying to distill it into something that I can describe to someone else.
Well, excellent. Along those lines as well, with a new set of targets potentially for you, I see that you were also awarded the Ellison Medical Foundation Senior Scholars Award in Aging. And I know there was a recent paper from Dr. Gareth Morgan in the UK about aging and myeloma. And so I'm wondering if your work in this area can also be applied to myeloma, or if you've looked at that at all.
I haven't looked at that. That's a very interesting connection that I haven't explored, and I'm not familiar with that work you cited. I should definitely look that up.
It's pretty recent, just in the last couple of weeks.
I'll just want to look that up.
So I guess a final question: how can patients help you do your work in the best way you can?
Oh, that's easy. It's the message I gave before, and that is we need grassroots support for more biomedical funding. I can't overemphasize this, in that this sequester has hurt us so much. I'm laying off researchers. I can't hire I can't buy the pieces of equipment that I need, and this was on top of already many years of anemic funding. This doesn't happen magically. The research tracks the funding. If there's more funding, there's more research. And so I would just encourage your listeners to let their voices be heard and to continue to make sure that our legislatures know the importance of basic research.
You've given us the marching orders. Now we have two ways patients can make a difference in setting the pace of cancer research. So one would be joining clinical trials, which is the reason we're doing this series. And what you're saying is that we can make a difference in helping support the idea of more federal funding for cancer research. I just want to mention that this is possible. And I learned about this from the book Emperor of All Maladies. In the 1940s, no one was talking about cancer research. And a woman named Mary Lasker, whose husband died of colon cancer, she sort of took it upon herself to decide that federally funded research needed to be in place. So she was very instrumental. She and her husband at the time were very instrumental in helping kind of reinvent the American Cancer Society and turn it into an organization whose mission it was to promote cancer research. She liked to say, if you think research is expensive, try disease, which I think we've all found to be true. And because of her efforts over many, many years, she helped expand the NIH budget from $2.4 million in 1945 to $5.5 billion in 1985. So it's a clear example that patients and caregivers can make a big difference in having federally funded cancer research. So thank you for pointing that out. Dr. Cruz, we are so grateful that you took the time to join us today. We are very fortunate to have your energy applied to the target of myeloma and hope that we can do multiple things to help support you in your valuable work.
Thank you very much, and I wish you the best.
Dr. Cruz performs his research as part of the Cruz Laboratory, but he's not actively treating multiple myeloma patients, and so he is not available for advice or questions. Thanks for joining us for another episode of Innovation on Myeloma on the Inpatient Myeloma Radio Show. Join us next Friday for another episode to learn more about how your participation can help push faster towards a cure.
It is Ryan here, and I have a question for you. What do you do when you win? Like, are you a fist pumper, a woohooer, a hand clapper, a high fiver? I kind of like the high five, but if you want to hone in on those winning moves, check out Chumba Casino. At ChumbaCasino.com, choose from hundreds of social casino-style games for your chance to redeem serious cash prizes. There are new game releases weekly, plus free daily bonuses, so don't wait. Start having the most fun ever at ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.